Welcome to today's webinar. Rapid Sterility Testing is presented by Andrew Taylor, ARL Biopharma Microbiology Lab Supervisor. Andrew has been with ARL since 2014. Prior to joining ARL, he worked as a research microbiologist at the Center for Disease Control and Prevention, CDC, in Atlanta, Georgia. Andrew received his Bachelor of Science in Zoology Biomedical Sciences from the University of Oklahoma and a Master of Science in Forensic Sciences from Oklahoma State University. We also have Brian Kelly, Director of Business Development, and Vince Bobbin, Laboratory Director, on the call to assist with questions after the presentation. Brian Kelly will also moderate the Q&A session. Today's webinar is accredited through ACPE and is worth 1 CE credit hour. CE credit is applied following completion of an evaluation link found on the last slide of the presentation handout. You can find this handout on the lower portion of the GoToWebinar toolbox under Handouts. We are recording this session and will include the recording and evaluation link in a follow-up email. Questions may be submitted today throughout the webinar using the GoToWebinar toolbox on the right side of your screen under the Questions tab. We will answer the questions submitted following the presentation. If we do not get to your question today, a list of questions and answers will be emailed to all attendees following the webinar. Thank you for joining today's session.
Okay, the topic of today's webinar will be rapid sterility testing. The purpose of this webinar is purely educational, where I'm declaring no conflict of interest. As Amy mentioned, this webinar has been approved for 1 hour of CE credit. And this slide includes additional information for the CE credit, including the ACPE course number. The goals of today's webinar are to define rapid sterility methods, to compare and describe some of the most commonly used rapid sterility methods, to explain rapid sterility method validation, and to summarize what multiple regulatory bodies, including the FDA, USP, PDA, and state boards have to say about the matter, to outline factors that a firm should take into account when implementing the use of rapid sterility methods, and finally, to highlight several of the benefits that rapid sterility testing has for compounding pharmacies. Rapid sterility methods are part of a larger group of tests termed Rapid Microbiological Methods, or RMMs. This term generally includes rapid sterility methods, but is a bit more broad, where it also includes bioburden testing, environmental monitoring testing, and so on. For this webinar, I will focus specifically on the RMMs that are used for sterility testing. So what is rapid sterility testing? These are tests which offer a faster alternative to traditional sterility testing, such as USP Chapter 71, as well as the European and Japanese compendial equivalents. Traditional culture-based sterility testing techniques require extended incubation times, which then take a relatively long time to produce a final result. Since timely microbiological data are often critical, not only for product release, but also for continuous process monitoring and control, rapid sterility testing is becoming more prevalent as well as useful. In order to reduce, or in some cases eliminate, the incubation times required, rapid sterility systems use modern technology and automation to detect the presence of microbial contamination. Many rapid microbiological method technologies have been developed over the last 20 or 30 years. Several of these have been marketed heavily in the clinical sector. However, some have specific pharmaceutical applications, where others have been developed solely for the pharmaceutical industry. Here is a list of various rapid microbiological methods organized based on the type of determination, detection method, and so on. Today I will be going into a bit more detail regarding some of the most common rapid microbiological methods that are used as an alternative to traditional sterility testing. Those are ATP bioluminescence, detection of CO2 production, and solid phase cytometry. As you can see, these three methodologies vary with respect to detection method, as well as the time to result. I will cover that in a bit more detail later in the presentation. Growth-based rapid sterility technologies differ from conventional culture methods in that they rely on the detection of biochemical or physiological growth indicators rather than visible or macroscopic growth. This generally allows for a much more rapid detection compared to traditional methods, but it requires a variable enrichment or growth stage before microorganisms can be detected. That is especially true in samples containing very low levels of contamination. The first type of growth-based rapid sterility testing methodology I will be discussing are those based on the principle of ATP bioluminescence. Adenosine triphosphate, or ATP bioluminescence, is a well-established rapid method for assessing contamination levels in pharmaceutical products and raw materials. Specifically, this method utilizes a substrate and enzyme combination, luciferin and luciferase, to break down microbial ATP from growing cells to produce visible light. This light is then measured using a luminometer, where the amount of light measured is relative to the number of microbial cells present. One of the main drawbacks of this technique is that care must be taken to minimize the presence of non-microbial ATP in a sample, as that can also be detected. The following slides include examples of systems which employ ATP bioluminescence. One commonly used rapid sterility system which utilizes ATP bioluminescence is the Milliflex Rapid platform. This is from EMD Millipore. Testing using this system entails the filtration of a sample, where then the filter is placed on an appropriate agar medium and then allowed the growth of microbial colonies after that. These colonies are then treated with an ATP releasing agent, followed by the addition of that luciferin and luciferase that I previously mentioned, where photons of light from each colony are then detected by the luminometer and a cell count is reported. With this technique, it is also possible to continue incubation to form larger colonies in case that subsequent microbial identification is deemed necessary. Some of the known strengths and weaknesses regarding the Milliflex Rapid system are listed in the table on this slide. As is the case with multiple methods I will be discussing today, one of the biggest strengths with this system is that the results are instrument generated, meaning they are objective and require no analyst interpretation. This system relies on the growth of microorganisms, which is considered both a strength and a weakness, in that it is more akin to a traditional sterility test, but the growth step does require additional time prior to results generation. Since the system does utilize a membrane filter, this does limit the type of samples that can be tested to only filterable products. And as mentioned on a previous slide, this method does allow for microbial identification in the event that an investigation is required. So the fact that the contaminant is not destroyed or lost is definitely considered a strength with the Milliflex Rapid system. A second common rapid sterility system, which utilizes ATP bioluminescence, is the Celsis Advanced platform. This is produced by Charles River Laboratories. Testing using the Celsis Advanced system is actually initiated exactly like a traditional sterility test, where a product is first processed using a suitable method, and that can be either membrane filtration or direct inoculation. The sterility test medias are then incubated for an appropriate amount of time. That would be based on the method validation, which we will discuss a little bit more in detail later. After appropriate incubation, aliquots of both media types are placed into small cuvettes and loaded into the instrument. The Celsis Advanced system then adds the luciferin and luciferase enzyme reagents, which catalyze the conversion of microbial ATP to light. One benefit associated with this system is it actually allows for full walk-away automation once those samples are loaded and the assay has begun. Here are some of the other recognized strengths and weaknesses associated with the Celsis Advanced system. Similar to the previous system discussed, the results produced by the Celsis Advanced are instrument generated. This elimination of analyst interpretation is very beneficial with respect to data integrity and consistency in methodology. As with other growth-based systems, testing performed using the Celsis Advanced is built on the premise of traditional sterility testing. But again, the enrichment stage does add time to the assay before a final result is generated. Unlike methods which require a sample to be filterable, one notable strength associated with the Celsis Advanced system is that it allows for great flexibility in sample types. Suspensions, oils, and really any sample that can be tested using traditional sterility methods can also be tested using the Celsis Advanced system. Adequate suitability testing must still be performed to ensure non-microbial ATP is absent from the sample, or that the sample properties do not slow or inhibit the enzymatic reaction that this technology uses. At this point, I would like to remind everyone to ask any questions you may have using the webinar control panel. We will be answering many of those at the conclusion of the presentation. The second type of growth-based rapid sterility testing methodology I will be discussing is that of colorimetric growth detection. This detection method relies on the color change being produced in a test media or assay materials as a result of microbial metabolism during their growth. This often specifically applies to CO2 production. This does limit the range of organisms that can be detected by this method based on their specific metabolisms. Systems are available which are able to detect most aerobic organisms as well as those acid-producing microorganisms such as lactobacilli or certain yeasts. One common rapid sterility system which utilizes colorimetric detection is the Bactalert 3D. This is from bioMérieux. This system was originally designed for clinical applications, but has also been found to be suitable for testing of some pharmaceutical materials. Testing using the Bactalert 3D equipment begins with samples being added directly to bottles of liquid culture media, which are then incubated inside the system at 2 different temperatures. During certain microbial growth, CO2 in a closed container accumulates and diffuses into the colorimetric sensor at the base of the bottle. Specifically, hydrogen ions interact with the sensor, resulting in a decrease in pH, causing the sensor to change to a yellowish color. One notable benefit with this system is that it automatically monitors all samples and all sensors every 10 minutes, which provides very up-to-date test data. On this slide, a list of strengths and weaknesses associated with the Bactalert 3D system are shown. Again, like previous growth-based systems mentioned, the results produced by this equipment are instrument-generated, which eliminates the subjectivity associated with manual reads. This system is unique in that it offers continual monitoring. As mentioned previously, it reads every 10 minutes, which is extremely beneficial with respect to real-time results. This allows for a much more timely approach to out-of-specification investigations or other potential contamination events. One notable drawback of the system, as previously mentioned, and other similar ones available, is that the colorimetric detection is dependent on the microbial contaminant's specific metabolism. This limits the range of microorganisms which can be detected using this technology. The last category of rapid sterility methods I'm going to discuss today are those which are non-growth based. These systems do not rely on microbial growth to detect contamination, but instead use cell labeling techniques to detect and quantify viable microorganisms. This approach has the potential to detect a wide range of our organisms within only a very few minutes because, again, it does eliminate that growth step. With that said, there are some systems which may require a slight or a small enrichment step where very low levels of contamination may be present. Generally, when we say very low levels of contamination, maybe less than 10 CFU or so. One of the most well-known examples of a non-growth-based rapid sterility platform is the ScanRDI system, again from bioMérieux. This platform combines methods of viability staining as well as solid phase cytometry. With this system, the test sample is filtered through a polyester membrane where any microorganism retained on the filter is then labeled with a non-fluorescent substrate. Within metabolically active cells, the substrate is enzymatically cleaved to release a fluorochrome, which is retained in cells with intact membranes. A laser scans the surface of the membrane within approximately 3 minutes, and viable cells are then detected. Based on certain system settings, autofluorescent particles, membrane fluorescence, and background noise are rejected, and a total viable cell count is reported. Many firms also include a step where positive results are manually observed using a phase contrast microscope. This is done for confirmation of potential contamination. The biggest strengths associated with the ScanRDI system is the impressive combination of speed and sensitivity, where detection down to a single bacterial or fungal cell can be performed in a matter of minutes or hours. When time to result is a priority, any system which does not require a growth step is hard to match. As with other systems which utilize a membrane filter as part of their method, the sample types which can be tested are thus limited to those which are filterable. Regarding results generation, because there is often an observation step to confirm contamination, results are somewhat more subjective compared to many growth-based systems which do not require analyst involvement. And finally, since no growth stage is afforded, microorganisms which may be present in the test sample are lost during testing. This means that should contamination not be homogeneously distributed in a product batch, it may be impossible to fully investigate or to identify microbial contaminants. Moving forward to one of the next goals of our webinar today is to discuss rapid sterility method validation. Perhaps the most important facet of employing a rapid sterility method is confirmation that a thorough method validation protocol has been completed. All of the systems mentioned during the previous slides have undergone full validations by their respective vendors, as well as numerous labs around the world. Since the validation is considered hugely important, and they can vary based on technology used as well as the lab's implementation of that technology, the FDA has not blanketly accepted any rapid sterility technologies as a whole. They are, however, open to reviewing validations of methods where they have been approved on a case-by-case basis. To address one question that often gets asked, that is, does my state board allow for rapid sterility testing? Currently, we are only aware of one state board that does not accept an alternative sterility method, and that is California. Historically, the task of implementing rapid sterility testing was considered very difficult and a barrier to entry for QC microbiology labs not familiar with alternative rapid methods. However, regulatory agencies have responded and have published additional guidance and documentation to assist implementation. Within the United States, PDA Technical Report Number 33 and USP Chapter 1223 can serve as great starting points for the validation of any rapid microbiological methods. USP Chapter 1223, titled Validation of Alternative Microbiological Methods, is referenced as an appropriate alternative by USP 797, which states specifically sterility testing must be performed according to Chapter 71 or a validated alternative method per Chapter 1223. The subsequent slides delve more into the parameters which have been assessed as part of a validation protocol based on the guidance in USP Chapter 1223 and PDA Technical Report 33. This slide shows Table 1 from USP Chapter 1223, which is titled The Validation Parameters by Type of Microbiological Test. As you can see, this table is broken down based on quantitative and qualitative test methodologies. Since sterility testing is a qualitative test, meaning that it yields a result of growth or no growth, or sterile or not sterile, we will focus mainly on the left column here. The specificity of an alternate quantitative sterility method is defined as its ability to detect a range of challenge microorganisms specific to that technology. A range of microorganisms can be defined in several ways, but at minimum, validation of a rapid sterility method should include the 6 organisms listed in USP Chapter 71, and it generally includes the addition of various other environmental isolates, organisms which have been identified from product failures, representative slow-growing organisms, and so on. Specificity is demonstrating by using an organism challenge level that is above the limit of detection, to be discussed in a moment, generally around 100 CFU of each microorganism, where both the compendial and alternative methods are performed with the expectation that comparable recovery is shown. The limit of detection, or the LOD, is defined as the lowest number of microorganisms which can be detected under the stated experimental conditions. To determine the LOD, suitable diluent solutions are inoculated with serial dilution range of each challenge microorganism from the panel previously defined. The level of inoculation should be adjusted to target approximately 50% of diluted samples that show growth in the compendial test. Commonly used CFU counts are approximately 5 CFU per test, approximately 0.5 CFU per test, and approximately 0.05 CFU per test. This is done again with the expectation that negative results will be observed using both test methods. Both the compendial and alternative tests are performed using a sufficient number of replicates to allow for appropriate statistical analysis. That's generally either the chi-square test or another appropriate statistical approach. Next is repeatability, which is defined as the degree of agreement among individual test results when the procedure is applied repeatedly to multiple samplings of the same suspension of microorganisms. It then also uses different suspensions across the range of the test to ensure the results are consistent and repeatable. Robustness, Robustness is defined as the capacity of the method to remain unaffected by small but deliberate variations in method parameters, for example, reagent volume, incubation time, or ambient temperature. Slight variations to these parameters provide an indication of reliability of the method and equipment during normal usage. It is important to understand that a measure of robustness is not a comparison between the compendial and alternative methods. Rather, it is a necessary component of validation of an alternative method so that the user understands the limits of the operating parameters of that method. Ruggedness is defined as the degree of precision of test results obtained by the analysis of the same sample, again under a variety of typical test conditions. For example, different analysts, different instruments, and different reagent lots. The expectation therein is that test results obtained by testing the same samples with varying analysts, instruments, or reagent lots should still be consistent. It is noteworthy that it is not uncommon for the end user to rely on data supplied by the test system manufacturer for both robustness and ruggedness determinations. When validating rapid methods for sterility testing, including stressed or injured microorganisms is highly recommended, as they are more representative of potential contaminants that may be present in samples to be tested. PDA Technical Report number 33 specifically mentions that a complete method validation should include the successful detection of stressed organisms. In order to perform this testing, methods are often used to obtain stressed or injured test cultures prior to testing. Some of these methods are to expose microorganisms to certain environmental conditions, such as UV, heat, cold, adjustments to pH, certain antimicrobial conditions, such as exposing them to disinfectants, or to put them through sublethal sterilization conditions. Either concurrently or prior to testing, these stressed microorganism cultures should be plated and enumerated to ensure reduction in viability. The expectation is that reduced CFUs should be present in the injured group compared to the control or healthy cultures. Testing is then carried out similar to how it was for previous validation tests, in that both the alternative and traditional methods are performed using a sufficient number of replicants to allow for appropriate analysis. Ultimately, the aim of validation testing is to demonstrate the acceptability of the alternate procedure relative to the current microbiological practice. To do that, the laboratory must demonstrate that the new procedure is as good as or better than the current procedure in terms of the ability to detect the presence of microorganisms. This is considered equivalence. USP states that alternate methods may be used if they provide advantages in terms of accuracy, sensitivity, precision, selectivity, or adaptability to automation. It also says that they must be shown to produce equivalent or better results than the referee method or the compendial method for any given quality attribute. Similarly, the FDA Guidance for Industry document Analytical Procedures and Method Validation, Chemistry, Manufacturing, and Controls Documentation states that a validated alternative procedure must be shown to have performance equal to or better than the regulatory procedure. When comparing two test procedures to show equivalence, statistical evidence is assembled to demonstrate non-inferiority or statistical non-inferiority. For example, with sterility testing, equivalence may be shown if there is no statistical significant difference between the two groups of results generated when performing testing with both the compendial and alternative methods. At this point, I would like to once again remind everyone to enter any questions you may have using the webinar control panel. The next slides cover some important things to consider when deciding if rapid sterility testing is right for you. First and foremost, it is important to always remember that patient safety is best served by detecting microbial contamination prior to a product's administration. In order to decide if rapid sterility testing is right for you, and further, what methodology meets your needs, several factors play into this risk-based decision. This includes time to result. As mentioned previously, methods which do not require a growth step offer the fastest turnaround time. If time to result is the most important factor, looking into one of these may be your best option. Second is specificity and sensitivity of the method. Can it detect the appropriate range of organisms such that you feel confident with releasing a product based on a sterile result? Next is sample size. Do you or does your testing lab have sample size requirements? Often this is still based on guidance from USP Chapter 71, Tables 2 and 3, but it is possible that labs have developed a justification for reduced sampling. Another point to consider are your specific product attributes. For example, many compounded sterile products and cell therapy products, due to their short beyond use dating or BUD, would benefit from an overnight test or at least one that is completed within 48 hours, where there are other products that may not require such a short time to result. Other factors to consider when deciding which rapid sterility test is right for you is the ability of the system to support aseptic material handling, meaning, is the system closed? Does it inherently reduce contamination based on its design? Or does it perhaps do the exact opposite, where increased false positive results may be seen? Next, if you're using your own lab or a contract testing lab, as with any analytical testing, it is important that you have confidence in the knowledge and experience of the group actually running the assay. As mentioned during previous slides, various methods allow or limit types of samples to be tested, filterability as an example. You must decide if, based on your production specifics, does this impact you or your ability to achieve rapid sterility results for your entire range of products. Lastly, I would like to note that just like with any thorough sterility test, method suitability testing is required to ensure a proper method is chosen for each unique product formulation. Sampling requirements will vary based on methodology and laboratory's requirements. However, it is important to note that this will be a needed step prior to performing release rapid sterility testing. Rapid sterility testing offers numerous benefits compared to traditional sterility testing. Most notably is shortened test times. The wait for a traditional sterility test result introduces risk into the production process in the event of a contamination. It introduces additional storage requirements, merchandise hold times, as well as delays to market. As noted previously, certain products with short shelf lives are prime candidates for testing using rapid sterility methods. A faster test time allows for improved efficiency across many facets of a production cycle, including storage and distribution. This can result in reduced waste due to products exceeding their BUD while sitting on a shelf or in a warehouse. These efficiency gains create further benefits by allowing firms to more rapidly respond to patient and client needs in real time. In addition to gains in efficiency and production, an extremely noteworthy benefit to shortened sterility test times is that it offers the ability to perform investigations in a more timely manner. Rather than waiting 14 to 18 days for a traditional sterility test, being given an under investigation or a not sterile result closer to the date of production allows for a more effective investigation, as well as the ability to implement appropriate corrective actions sooner. With a faster response comes a faster recovery following any contamination event. The benefits of rapid microbial detection extend beyond just a faster time to result for quality control testing. While a faster result is a major positive impact of rapid detection, users of many systems also gain the ability to address and meet data integrity expectations and requirements from governing agencies. With many of the technologies discussed in this presentation, the implementation of a rapid sterility method replaces the subjectivity of a traditional visual confirmation with an automated reagent-based assay controlled by an instrument. Results are controlled and reported through the software, avoiding transcription and interpretation errors potentially caused by an analyst recording or reporting handwritten results. Instrument usage logs can also be generated, which can help to meet audit trail requirements. The picture on this slide shows 4 separate traditional sterility test broths, each demonstrating a different visual appearance. In this specific case, the 2 samples on the left are sterile, and the 2 on the right are contaminated. While the traditional sterility test method is able to overcome this issue by subculturing any turbid media, this highlights the fact that the use of only visual observation may lead to inaccurate, or at the very least, delayed results. To summarize what we've discussed today, rapid sterility testing is a group of methods which are part of a broader group termed rapid microbiological methods, or RMMs. Rapid sterility methods are alternative test methods to use in place of or in conjunction with traditional sterility testing, USP Chapter 71 as an example, which allow for a shortened or in some cases no incubation times. These methods use advanced technology to detect microbial contamination, which allows for a faster test time, and since the user does not have to wait for visible growth to appear. Numerous vendors have created rapid sterility testing systems which employ different modes of detection, growth versus non-growth based, for example, in order to provide an alternate to traditional sterility testing. These systems differ with respect to testing time, meaning whether a growth or enrichment step is necessary, product types able to be tested, for example, filterables or non-filterables, subjectivity of results, are they instrument-based or does it require analyst input, and finally, the ability to further investigate out-of-specification results. Is the microorganism able to be identified once contamination is expected? These are all factors to take into account when deciding which rapid sterility testing system best meets your needs. Rapid sterility method validation is a multi-step process with the ultimate goal of demonstrating equivalence, or in some cases superiority, of an alternative test method to a compendial test method. As noted, there are multiple resources available to assist in the design of a validation strategy, with the notable recommendations and guidelines being produced in USP general chapters, FDA guidance to industry, and PDA technical reports. Again, to reiterate one final note regarding validation testing, that is that method validation is completely separate from formulation-specific method suitability. Once a rapid sterility test method has been validated by the lab that plans to use it, each formulation to be tested still requires method suitability be performed in order to demonstrate its compatibility with the system or the technology. The use of a rapid sterility test is a risk-based decision where the stakeholders must bear in mind that patient safety is paramount, that good manufacturing practices and superior quality system management must be maintained, irrespective of whether you change to a rapid sterility method or not. Multiple factors, including time to result, specificity and sensitivity, product applications, sampling requirements, as well as knowledge and experience of the testing lab, must be taken into account when considering the implementation of rapid sterility testing. Rapid sterility testing offers multiple advantages compared to traditional sterility testing. Most notably is shortened test times. This creates benefits across an entire production schedule, including the release of products faster. With a faster test time comes the ability to perform investigations and implement corrective actions in a more timely manner. Additionally, reduced subjectivity in results, as well as improvements to a firm's data integrity compliance, are both benefits associated with moving to a rapid sterility test method. And as a final summary, time is money. A faster test time allows you to release products faster, reduce inventory and improve warehouse utilization, shorten production cycles, respond to, and importantly, recover from contamination faster, and finally, to improve overall efficiency. Here's a list of references that I compiled in preparation for today's webinar. And we will now transition to the Q&A portion of the presentation, where our panelists and I will address questions that have been asked. Thank you very much for your time today.
Okay, thank you, Andrew. Thank you to all the attendees. We've had several questions submitted throughout the presentation. Myself, Brian Kelly, the presenter, Andrew Taylor, Vince Bobbin, our laboratory director, and Dr. Kupik are available here to answer some of the questions. We will go through some of them during this broadcast, and we'll answer all of them in a Q&A document that will be sent out to all attendees within the next week or so. Okay, one of the first questions, Andrew, I think you'd be appropriate for this one. Because the BACT alert uses CO2 production as its detection method, does that mean that it may not catch all of the organisms that a 71 would catch?
It is possible. Because it does rely on CO2 detection specifically, as mentioned during the presentation, that does limit the ability to detect all classes of organisms, where USP 71 uses those 6 specific classes to represent basically multiple different types of contaminants which may be present. So it is possible that CO2 detection limitation will limit those organisms as well.
Okay. One of the questions we have is, can you comment on the cost of initial setup, meaning method suitability, and routine analysis? I'll take that one. So as Andrew mentioned, the cost or the technology is very considerably. So the cost would be based upon on the method. Some laboratories offer some tests, some laboratories offer other tests. For us, as we're using the Celsus platform here, as Andrew mentioned, the front half of the Celsus test is exactly the same as a 71 test. So we still incur all of those same costs with closed membrane filtration systems. And then as the endpoint, instead of the end result being obtained by a visual observation, we must then use the Celsus system, which then uses the reagents and things. So the cost is going to be higher because the costs for the laboratories are higher on that particular test. But just need to check with your laboratory, and they should be able to provide some routine pricing on that. Another question, why are including stressed organisms important during the validation?
Yeah, I can speak to that. I touched on that slightly during the webinar presentation. But if one thinks about likely potential contaminants in a compounded product, generally speaking, they're not going to be in their healthy or growth phase, and they would be, stressed or quote injured based on perhaps the pH of the product, the temperature at which it was shipped or stored. So again, to reiterate, recovery of stressed or injured microorganisms is going to be much more representative of anything that may be present in a sample to be tested for sterility.
Okay. One of the questions is, can you compare the Celsus system to that of the scan RDI? I think one of the important things to talk about here would be the potential for out-of-spec investigations and how that might proceed. I think what you said in your presentation was that the scan RDI is a destructive test. So say that someone was to obtain a non-sterile test result, what kind of out-of-spec investigation can occur if there is no sample?
Well, obviously, any out-of-spec investigation should include more than just a potential microbial ID of the contaminant. It should assess, every bit of the process that led to that potential non-sterile result. So a firm could still obviously take a look at various other in-process checks that they have. Of course, the lab performing the sterility test would have various controls in place that they could assess to ensure that they're confident in the result that they generated. But one important piece of a sterility out-of-spec investigation oftentimes is that identification piece where the scan RDI system does not allow for that as some of the other growth-based systems do.
Okay, good. Next question, growth-based rapid sterility tests utilize 2 to 3 days incubation period, question mark?
That can vary based on method and also based on that individual facility's validation protocol. So one can imagine if somebody designed a validation protocol to only use healthy organisms at relatively high CFU counts, say 100 CFU or even less than 100 CFU, but greater than 10, 2 to 3 days would probably be plenty, frankly, with a healthy organism. If you then incorporate very low levels, like we spoke about in the LOD portion, limit of detection, and or stressed microorganisms or injured microorganisms, which do not hit that growth phase as quick as healthy or happy organisms do, 2 to 3 days may not be sufficient in those cases. So it really will depend on the specific validation protocol, as well as the specific methodology that a lab is using.
Okay. Another question related to CO2 detection and specifically the Bacti alert systems, what you talked about. Given the limitation on detectable organisms, can a CO2 detection method be used as a single method to meet 71 requirements?
Well, without going too far into the first portion of that question with regard to it being a CO2 detection method or otherwise, keep in mind that none of the rapid methodologies we said today satisfy USP 71. They are to be considered alternative test methods that can be used in place of, or again in conjunction with USP 71. But whether it be growth-based, such as the CO2 detection, non-growth-based, such as the scan RDI system we mentioned, none of those actually satisfy USP 71, which is very clear on how long to incubate samples. It specifically tells you exactly how long to do that.
Okay. Okay, one of the questions is a ARL specific question. Will a method suitability library be available to customers? So we currently have a process where we will compare incoming new formulations to formulations in which we have already established method suitability. We are currently only utilizing that methodology for USP 71 or traditional growth-based sterility tests. At some point down the line, we may develop a library system for the Rapid, but it's not something we're offering at this time. Several questions about Andrew's comment about the state of California not accepting rapid sterility test methods. It's been a few months or something since the last time we checked in with the California Board of Pharmacy. That was their stance whenever they developed those guidance documents about compounding in the state of California. So as far as we're aware, they have not changed their stance on that. I think they were planning on adopting the new 795 and 797 versions when those were finalized, but as everybody on this call knows, that did not occur. So California Board of Pharmacy is, as far as we know, is still continuing to use those guidance documents that they developed several years back. Okay. How many organisms are required for method suit, and is this the same number of organisms as required for 71?
Well, a requirement is going to be definitely based on a laboratory's protocol. It is definitely recommended to, again, use at minimum the 6 compendial organisms listed in USP Chapter 71. In the validation strategy or the validation protocol of the method. And then when we're talking about method suitability or the product formulation specific suitability testing, it is reasonable to use, again, the 6 USP 71 challenge organisms to ensure that the growth would be comparable to a positive control using that alternative test method in the presence of that product.
Okay. Thank you. Okay. Well, I think that covers all of the questions that were submitted. Feel free to reach out to us should any further questions come to mind. Thank you, Andrew.
Thank you all for attending today's webinar. If you need CE credit, please fill out the evaluation link found on the last slide in the handout. There's also a QR code that you can scan that will send you directly to the survey. This evaluation is required to obtain 1 hour of CE credit. Thank you very much and have a good day.